So cancer is another condition that affects and plagues pretty much the whole body. Right? It affects the organ system's ability to maintain homeostasis, and the word homeostasis means it's our natural rhythmic balance that we want to be at. And unfortunately, whenever you have a detour and you deviate from homeostasis, then you have disease. This cancer can be literally found in any system, any tissue of the body. Out of all the tissues that you see here, so you have different kinds of epithelial, muscle, nervous tissue, connective tissue, the most common tissue type that gets damaged tends to be epithelial. Epithelial is like your skin because it's oftentimes exposed to the environment. It's not to say that the other kinds of tissues are completely immune. They can also be damaged because of mutation, because of environmental factors like we said on Tuesday. But you can see that all these systems, whether it be endocrine system, respiratory system, urinary system, lymphatic system, integumentary system, the skin, muscular system, digestive system, cardio, and even nervous tissue can be affected. And it all comes down to the number of cells that grow and divide. And when you grow to a point and divide to a point where the cells are not able to function, then that's generally going to be cancerous and problematic. So we had a chance to look at a question, and if you didn't get a chance to answer this question, go ahead and respond. So cancers are named based on which of the following. This was a quick question that we went over. So go ahead and answer this, and then we will keep the ball rolling. All right. So this was the one that I think I showed you guys on Tuesday, but we just closed it there. So please answer. 82 people answered. That means there's a few of you guys that didn't get a point. So go ahead and respond so you get the points for this week or today. So one of the key concepts to cancer is diagnoses, is the ability to detect them early on. And there are certain kinds of warning signs that you could look for. It could be including of changes to the way your bowel movement works, if you're having bleeding, if you're having some kind of injury that doesn't heal, if you can physically feel lumps on your body. So there are some things that you can do to assess whether or not you might have some early signs. And of course, routine screening, like using different kinds of self-examination, pap smears, rectal exams, colonoscopies, sigmoidoscopies, different ways of assessing the internal condition of your body. Right? So that's really the trick is to have these routine checkups and to run these tests to see if there are any issues. All right, good. So the answer for this one, you see, is the type of tissues or organs they originate from. So 76 of you guys got that. All right, so that's good. Awesome. Great answer C. Great job. Okay, so let's go ahead and do a few slides first. So what I wanted to do is I wanted to give you a little bit more content. We'll come back to the discussion. So really the discussion of cancer is all about the cell. Right? The cells, like we have said, are these machinery in our body that comprise of the tissue. And they are able to acquire materials and energy. They're able to respond to the environment, the fluid content, the salt content. They reproduce, they then divide, they grow. And cells, like we've said before, could be either simple, right, or true cells, eukaryotic or prokaryotic. And that all depends on just the nucleus. Right? Just a quick recap is the prokaryotic cells are ones that do not have enclosed nucleus, whereas the eukaryotic cells have this kind of encapsulated, rounded envelope nucleus like you and me. And so when a cell divides, it goes through a process called mitosis. Okay, so I want you to think about this. So mitosis is a very, very natural, good process of cell division. Right? Cell division involves a lot of machinery. It involves a lot of different kinds of organelles to play its part and to split one cell to two cells. However, in cancer, that's when cell division is on steroids. It is that the cell division keeps on ramping up and it doesn't have any kind of sign of slowing down. And that becomes an issue. A few things to think about here is that there are common structures in all cells, and we can kind of compare that in reference to human cells, right? So I want to related to you and me because it's more tangible. When you learn about your own body, at least you know about it's your own system. So the nucleus is where it contains and houses the genetic information, and this is where all the DNAs occurred. Right? So this is what tells the cell what to do. So if anything were to get into the nucleus or damage that, then you can risk having mutations. The ribosomes are the machinery by which the DNA is converted to a message called the mRNA and then translated into a protein. So that's the machinery that makes a protein, or the output. We spent some time talking about the mitochondria already. That's the energy powerhouse of the cell. And then there's also structures that allow you to transport these different products. And these are the ER, endoplasmic reticulum, lysosome, and Golgi apparatus. These three are able to process the proteins or lipids, digest different kinds of nutrients and waste, and then ship them out. So think of the Golgi apparatus like the UPS or the FedEx. Right? So when you have a package and you want to mail it back out, or you're doing a return to Amazon, which I do too much of, you can go to the FedEx and then drop it off. Right? So it's a distribution center for the cell. And then, of course, we have the membrane of the cell, which is the outer boundary that allows items to go in and out. And the centrioles, which we haven't spent any time talking about, are these important machinery, these important protein-like fibers that will align the cell during cell division. So it guides what part of the cell to split. And that whole process is very, very fine-tuned because you don't want a cell to be split into a non-even way. Or you don't want one cell to have two nucleuses and one cell to have no nucleus. So let's talk a little bit about Spell's end goal. So cancer cells tend to have this condition of being non-specialized, whereas normal cells, healthy cells in our bodies, are specialized, which means that during development, based on genetic information, they are allowed to have a fate. So one cell that is the grandfather cell of them all becomes a progenitor cell, and over cell division, they can become either A, the neuron, B, a skin cell, red blood cells, heart cells, pancreatic cells, lung cells. They have this fate of differentiation. Cancer cells oftentimes ignore this entire genetic information, and they lose their function, and they also have abnormal nucleuses. So I'm writing this out here as some of the key traits. So if you ever were to spot a cell, a cancer cell under the microscope, they tend to have this distorted, uneven nucleus. So literally the shape of that round purple circle, that nucleus, is not the right shape or the size or density. And so when they have this abnormal nuclei or nucleus, that's also one indicator of cancerous cells. Apart from having this abnormal nuclei, in general, they don't divide into any specialized cells. So there's this lack of differentiation. Okay? And so what happens here is that cells are usually pretty specialized. They have a dedicated shape, a function, a goal, which also means that their reactions and ability for them to utilize enzymes and proteins are extremely specific. And going back to what we talked about in metabolism, right, where we said how enzymes require a substrate, right, some kind of a cofactor to bind onto that blue enzyme on the bottom. And if you have the right match, right, so this orange substrate has the right exact shape that matches that contour of the enzyme, then you have this reaction that can occur. And if you have something that's not the right shape, then this reaction wouldn't occur. And so what happens here is that in some cancers and some issues is that when you can target development of cancers, you could have drugs that might block an enzyme from doing its reactions so that you can slow down cancer. So that's why there's a lot of options to looking at treating diseases. Are you trying to slow down the reaction? Are you trying to compete with what normally will bind? Are you also trying just to cut off the cancer and to burn it off, physically take it away? There's a lot of ways to look at it. And one of the scariest parts about cancerous cells is that these cells have the unlimited amount of potential to divide, which means that they're considered immortal, okay, which is pretty crazy. So you could think of cancer cells as cells that can never end and constantly divide, or otherwise they're immortal cells. There's a book that has been a pretty good read if you're interested. It's called the Henrietta Lacks. It's the immortal cell line, but the study on how the first set of cancer cell studies began in our country. And it was really early on where there was a female patient that had this uterine cancer in her uterus, and her uterus would constantly divide. So the scientists, and this is way before they had the Bellman report and some of these ethical research protocols, so they actually took some of her uterus cell line and grew it, right? And obviously this was without her consent, so obviously it's an infringement on your rights, but they then froze her cell line, and then most of the cancer research right now are done on her cell line. So it's really a crazy story, but basically it's just, back then regulations weren't really that well put together. They took her cells for study, and obviously right now they're trying to get some royalties and some monies from that because of the contribution of her cell lines to research studies. So it's interesting to think about, but they're considered immortal, and immortal just means that they are able to divide over and over. They have unlimited number of times, whereas usually in human cells, they have these structures called telomeres that shorten every time a cell's divide. And there's this portion on a cell called the telomerase, which basically prevents the cell from losing its ability to divide. So it's like a little valve. And in cancer cells, you don't have that valve, right? So it's like there's this unlimited potential to divide. So information in all cells are stored inside a region called the nucleus, and they are stored in a form called DNA. You might have heard this before. DNA stands for deoxyribonucleic acid. This is a series of bases that then match up to create a code. There are 4 bases that code life. All right, so this content that I'm sharing with you are really non-disputable. This is all in the textbook. It's widely accepted everywhere. It's not current research. This is stuff that we've already accepted. So the 4 bases are, we have A for adenine, T for thymine, G for guanine, C for cytosine. And I want you to look at the picture on the bottom left. You can see that the structures are a little bit different, right? So the adenine, shown here in orange, has 2 rings, and it has a couple of nitrogen bases in there. So a couple of N's in here, a couple of H's in here. But the main thing is you can see 2 rings, right? There's 2 rings. There's a pentagon right here, and then a hexagon in the bottom, right? So for the chemistry people in here, whenever you see an empty angle, that represents a carbon. And for this guy, the adenine is a double base ring. On the top, guanine is also a double base ring as well, right? It has 2 rings, whereas cytosine and thymine only have 1 ring. You guys see that? It's 1 ring only. So for our purposes of Bio 10, just be aware of which bases are double ringed and which bases are single ringed. That's really the take home here. And because some of these bases have different numbers of rings, it might have a different property. I also want to show you that these rings are linked up together via a hydrogen bond, right? So you can see in the middle, this dotted line represents hydrogen bonds that occur between the 2 bases. So these bonds are not super strong. But when you have a ton of them, right, like the picture over here, a ton of these rings linked up, then you create a relatively stable structure. It's not extremely stable, like temperature and pH and acidity can damage DNA. But in general, right, it's relatively safe, it's relatively stable, and that's also why when you are providing any kind of transportation of DNA or RNA, it should be in a relatively set temperature. You don't want it sitting inside a car that's 100 degrees. You'll also notice over here that in every single one of these bases, you have 3 things, 3 anatomical features to a nucleotide. We have a nitrogen base, that's the upper blue C. We have a sugar, a pentose, shown here in green, pentose, and then a phosphate. So there's 3 parts to every single nucleotide, and they then link together like so. Right? You can see that right here. They link together and they form one little part of DNA, and the coil just keeps on growing and growing. When you read DNA, what happens is you're like, hey, how do I look at DNA? How do I start from the beginning to the end? Well, DNA is read from a 5' to 3' end, which means you have a region that has the 5' carbon, and then you end with a 3' end, right? So you have the start and finish. So basically, you read it from one direction down, and then you have the other strand going back up. So it's reverse. And the bases that pair together, the rule that links DNA together is as follows. A always goes with T's, and G's always goes with C's. One slight difference, one minute difference, is that DNA gets converted to RNA eventually as a message. And then in RNA, we have, instead of thymine, we have something called uracil. So it's a base change in RNA. All right, so that's a lot of information. You're probably a little confused. You're like, uracil, thymine, what's going on? So all you need to remember is that there is a base pairing rule in DNA, which is A to T and G to C. All right, so that's literally the code of life. You can't change that. It's just A's love T's and G's love C's, and that's how your DNA is linked and built together. So how does this relate to cancer? So I want you to think about this next couple of lines that I'm going to explain. Is that when the A is supposed to pair with T and G is supposed to pair with C's, let's say you have some kind of chemical or radiation or toxin comes in and damages the bond. Right? So that comes into your DNA and it messes up this bonding. So now A pairs with C. Now you have a problem, right? So essentially what happens is that when you have a number of these pairing that messes up, you then have a risk of mutation, and if it's the bad kind of mutation, then you can have cancer. That's a simple way of thinking of it. So just remember, A pairs with T, G pairs with C, and in order for these nitrogen bases to then store genetic information, all of this information is linked together and packed into what we call the genome. Right? This is a set of codes that will have different regions that will code for different instructions. And it's really challenging to look at this if you want to think of it linearly, because not one strand might code for one thing. It could be a part of this DNA and part of that DNA combined together might express some kind of protein. Right? So there's a combination of reads in your DNA. So genetic expression is a key concept of biology, and this is exciting because this is how you can understand how the mRNA vaccine that was quickly developed came to be. Right? So somebody basically provided the genetic code of DNA, and they took the DNA and then literally copied it into mRNA, and now it created this mRNA vaccine that could be then delegated and implanted into the cell, and the cell will take that in. So this is the storyline. The storyline begins that our cells have a nucleus, our cells contain DNA, and that DNA is fixed. Right? That just tells you what you are, who you are. A's pair with T's, G pair with C's. As you can see, this is the pairing rule. The second phase of expression is going from DNA to RNA. RNA stands for ribonucleic acid, and the first kind of RNA that we have here is called mRNA. The letter M just stands for message. All right, think M is message. It's the intermediate message between DNA and protein. And what now it does is that it takes the DNA, it reads what it has, and then copies it into the RNA form. The only difference here is that instead of having thymine in the A to T base pairing rule, you have uracil. So uracil takes place of thymine. So it's just a slightly different base, and that's all you need to remember. Right? There's a total of 5 bases, 4 in DNA, and then 4 in RNA, and then there's that one that switches around, right? So 5 total. And then after the mRNA gets generated, you can see that's still happening inside the nucleus. This is everything above this purple line, or this red line, is inside the nucleus. And then the final stage of genetic expression is taking the mRNA, moving that into the ribosome, which is a big red globular protein right here. It's machinery. And then that will then read the RNA and create a protein via translation. Obviously, this is quite simplified, but let me just summarize the 3 main points. Number 1, DNA is the instruction. mRNA is the message or the intermediate phase. And then protein or translation is to make the final product. Number 2 is going from DNA to RNA, we call that transcription. It's just a fancy word for that. And then going from mRNA to protein, we just call that translation. All right, so don't get confused, transcription and translation. The last point to remember is that in RNA we have uracil, and then in DNA we have thymine. All right, so those are some of the guiding rules to this part of the discussion. All right, so let me backtrack to our discussion for the day. So I want to get this discussion in. Now that you've heard a little bit of content, got a little bit of information in your mind, let's go back to this question. So describe how cancer is a physiological disease that occurs at the cellular level, and describe what you know about the term cancer. Right. So now that you've heard me talk a little bit about some stuff, you maybe have a lot more knowledge than I do about cancer. Maybe you know it more personally. Feel free to share, but go ahead and post your answer. I'm going to break you guys out to groups for a little bit over a minute, and we'll come right back. All right. So we have 5 more minutes. I want to get a discussion in for this week.